Welcome back to this next video, and in this video we are going to talk about the protein kinases and the different types of the protein kinases. So the first thing is what are protein kinases? These protein kinases they are actually enzymes that regulate the biological activity of proteins by phosphorylation of specific amino acids. Now these protein kinases, when they phosphorylate the specific amino acids in this protein, these kinases are using ATP as a source of the phosphate group. When these kinases they add phosphate group to specific amino acids in their target proteins, this addition of the phosphate group is inducing a conformational change from an inactive to an active form of the protein or vice versa. Now, what this means is that the protein kinases can do both of the things. They can actually they can actually convert a protein from the inactive to the active form, or they can actually move them from the active to the inactive form. If you see this, if this is a protein, or these protein kinases, when they act on this protein, they are going to convert in ATP into the ADP, and the phosphate group that it gets from the ATP is then added to specific amino acid in the target proteins. If you talk about the human genome contains about 500 protein kinase genes, and they constitute about 2% of all human genes. Now this means that these kinase gene, if you look at their percentage in the human genome in terms of the genes, they are playing a very important role. Now these protein kinases, they are also found in bacteria and plants, and they are playing their important role in the cellular signaling pathways. Now, up to 30% of all human proteins, they may be modified by the kinase activity. By that I mean they can be activated or they can be inactivated by the kinase activity. And these kinases, they are known to regulate the majority of the cellular pathways, especially those involved in the signal transduction. Now there are different kind of the kinases when you talk about the living organisms, and they are broadly divided into 4 groups. The first group of the kinases, they are known as the serine threonine kinases. The second type, they are known as the tyrosine kinases. The third one are known as the dual specificity kinases, and the last group that are known as the histidine kinases. Now, if you look at the name of these kinases, the amino acid is actually mentioned in the names of these kinases. So, if you talk about the serine threonine kinases, that means they are adding a phosphate group to the serine or the threonine amino acid. If you talk about the tyrosine, they are adding a phosphate group to the tyrosine amino acid. And if you talk about the dual specificity kinases, they can actually add a phosphate group to the serine, threonine, or the tyrosine. And the histidine kinases, they are adding a phosphate group to the histidine amino acid. We will discuss that in a while. If you look at the 4 amino acids that we are talking about, the serine, the threonine, the tyrosine, or the histidine, these are the structure of the serine, threonine, the tyrosine, and the histidine. So the common thing between these amino acids, that all of these amino acids have got hydroxyl group. If you look at the threonine, they have got a hydroxyl group over here. They have got a hydroxyl group over here. The serine have a hydroxyl group. The histidine, they have got the hydroxyl group, as well as the tyrosine, they have got a hydroxyl group. This means that the attachment of the phosphate group by the kinase enzyme actually needs the presence of a hydroxyl group. If you talk about the first class of the kinases, which are known as the serine threonine kinases, so what these kinases they do, that they are going to phosphorylate their target protein at the serine or threonine amino acids. These kinases have got the potential to attach a phosphate group both to the serine and the threonine. And the serine threonine kinases, this family is the largest of the kinases, and at least 125 of the 500 plus human protein kinases, they are actually the serine threonine kinases. If you talk about the chemical reaction that is performed by the serine threonine kinases, you can simplify it like there is an ATP molecule, there is a protein. So when the kinase act on this, the ATP is converted into the ADP, and the protein is converted into a phosphoprotein, because one of the phosphate group that has been removed from the ATP, which has converted it into the ADP, that one phosphate is added to the protein, converting the protein into the phosphoprotein. So the two substrates, they are the ATP and the protein, and the two products, they are the ADP and the phosphoprotein. If I give you an example of the serine threonine kinases that is there in the human being, so one of the important example is known as the RAF protein, which is actually the rapidly accelerated fibrosarcoma protein, or it is also known as the MAPKK. And this is one of the important protein of the MAPK signaling pathway. I have a detailed video on the MAPK signaling pathway, and I'll share the link in the description. So this RAF is actually a serine threonine kinase. If this is, say for example, serine, this is the structure of the serine amino acids. So when a phosphate group that has been added to it, it has been converted into the phosphoserine, and the protein has been converted into a phosphoprotein. Similarly, when you talk about the threonine, so when a kinase act on the proteins and they add a phosphate group to the threonine has been converted into the phosphothreonine. You can see the structure over here, and the protein has been converted into a phosphoprotein. The second class of the kinase, they are known as the tyrosine kinase. I hope you are getting the idea of what the name of the amino acid in front of the kinase term means. So when you talk about the tyrosine kinases, they are the kinase enzyme that can transfer a phosphate group from an ATP to the tyrosine amino acid of a specific protein inside a cell. So if you compare it with the serine threonine amino acid, so the amino acid is changing, so is the name of the kinases. So these tyrosine kinases are actually adding a phosphate group to the tyrosine amino acids in their target protein. Now the addition of a phosphate group to the tyrosine in the specific protein, that can actually work as a switch, an on and off switch in many cellular functions. If this is a tyrosine, and when a kinase act on this tyrosine has been converted into the phosphotyrosine, and the protein has been converted into a phosphoprotein. If you look at the famous example of the tyrosine kinases, one is known as the SRC protein. I have a detailed video on the function of the SRC, and I'll share the link in the description. In that particular SRC pathway, I have also talked about the focal adhesion kinases. Another important protein is known as the ABL, and another important one is known as the Janus kinase, which is actually one of the important kinase when you talk about the interferon signaling pathway. So these are just some of the examples of the tyrosine kinase that they are there in the human beings. Another class of the kinases, they are known as the dual specificity kinases. So if you focus on this term, it have got the dual specificity. What this means is that these dual specificity kinases, they are kinases that have the ability to add a phosphate group to both tyrosine, the serine, and the threonine. This means that the dual specificity kinases, they have the ability to add phosphate group to the serine, threonine, and the tyrosine amino acids. One of the important example of the dual specificity kinase, that is known as the MEK, which is the mitogen activated protein kinase, or for short that is known as the MAPKK. So if you look at my video on the MAPK signaling pathway, all of these proteins, they are there, and their functions have been discussed in detail. So if you talk about the dual specificity kinase, they have the ability to add a phosphate group to the serine amino acid, converting it into the phosphoserine. They have the ability to convert the threonine into the phosphothreonine, and they have the ability to convert the tyrosine in the phosphotyrosine. So therefore they are known as the dual specificity kinases. Another family of the kinases, they are known as the histidine kinases, and the histidine kinase, that is actually an enzyme that can transfer a phosphate group from an ATP to the histidine amino acid of specific proteins inside the cell. So these the name of these amino acids in front of these kinases are actually showing the substrates of these kinases. So in the histidine kinases, the histidine will be converted into phosphohistidine by the addition of a phosphate group from the ATP molecule. Now, what phosphorylation can do, as I've told you, that they can actually turn on and they can actually turn off a particular protein depending on the need of the cell. I'll be using the cyclin dependent kinase 1 as an example to show you how the phosphorylation is responsible for the activation or for the inactivation of the proteins. I've chosen this cyclin dependent kinase 1 as an example because this is one of the protein where both of the phosphorylations they are there. There are phosphorylation at different positions which make this protein inactive, and there are positions in this particular cyclin dependent kinase 1 which make it active. So let us discuss this in detail. If you talk about the cyclin dependent kinase 1, so this is one of the important protein in the cell cycle, and the CDK1 it actually combines with another protein which is known as the cyclin B, and it makes the CDK1 plus the cyclin B, it actually makes an mCDK which actually drives the M phase of the cell cycle. Now this CDK1, as the name indicates, this is cyclin dependent kinase. That means if the cyclin is attached to the CDK1, this will be in the active form, and if the cyclin cannot interact with this CDK, this means that the CDK1 will be in the inactive form. Now what happens is when the activity of the CDK1 that is not required, for example if you are in the G1 or the S phase of the cell cycle, so of course you do not need the activity of the cyclin dependent kinase 1 because its activity is required in the M phase of the cell cycle. So in those particular cases there are two important positions in the CDK1 which are phosphorylated. One of the position of the CDK1 is the threonine 14, and another important amino acid at position number 15 is the tyrosine. So the threonine 14 and the tyrosine 15, both of these they are phosphorylated by a specific protein, a specific kinase which is known as the V1. So the V1 is going to phosphorylate the threonine 14. It is going to phosphorylate the tyrosine 15. So you can put the V1 in the dual specificity kinases. Both of these phosphorylations, they are known as the inhibitory phosphorylations. What I mean by that is that if both these positions they are phosphorylated, this phosphorylation is going to inhibit the activity of the CDK1. So in this particular case, the phosphorylation of a protein is actually inhibiting the activity of this particular protein. Now when these two positions, the threonine 14 and the tyrosine 15, they have been phosphorylated by the V1, how it is going to inhibit the activity of the CDK1. These two phosphate groups, they are going to change the CDK1 into a conformation that it is not able to interact with the cyclin B, and if the CDK1 it is the cyclin dependent kinase and it cannot interact with its cyclin, this means that the CDK1 cannot be activated. So these two phosphorylations, they are inhibitory in nature. There is another protein which is phosphorylating the same CDK1, and that particular kinase is known as the CDK activating kinase. This CDK activating kinase is actually phosphorylating the position number 161 of the CDK1, and at the position number 161 there is a threonine amino acid. Now when there is a threonine amino acid at position number 161 in the CDK1, this is phosphorylated by the CDK activating kinase. This phosphorylation is actually stimulatory phosphorylation. What this means is that this phosphorylation is going to make the CDK1 in the activated form, or it is converting it into the activated form. Now if there are both inhibitory phosphorylation and stimulatory phosphorylation, what is the end result of this? Both of these phosphorylations, they are there, but as there are both inhibitory phosphorylations and there are that is going to change the shape of the CDK1 into a conformation that it cannot interact with the cyclin B. So what you do is that if you want to activate the CDK1, first you have to get rid of these inhibitory phosphorylations. There is a particular phosphatase. Phosphatases are enzymes that have the opposite activity of the kinases. Kinases are adding a phosphate group to a particular protein. The phosphatases are removing the phosphate group from particular protein or amino acids. So there is a particular phosphatase which is known as the CDC25. So the CDC25, what it do is that it removes the phosphate group from the tyrosine 15 and the threonine 14. This means that the function of the CDC25 remove these inhibitory phosphorylations, and when these inhibitory phosphorylations they are removed, this stimulatory phosphorylation is actually enhancing the interaction of the CDK1 and the cyclin B, and when the CDK1 and the cyclin B they interact with each other, the CDK1 is going to move into the active form and it is going to perform its particular function that are required in the M phase of the cell cycle. So if you like the video, please subscribe to my channel and share the links with your friends and your colleagues. We will continue the discussion in the next video.